Good morning, one and all. It is my pleasure to warmly welcome our distinguished speakers, respected faculty members, researchers, industry experts, scholars, and participants to the day 3 of our 5-day International Faculty Development Program. Over the past 2 days, we have explored exciting developments in the fields of artificial intelligence, biomedical imaging, and drug delivery systems, biomaterials, regenerative medicine, biomechanics, biosensors, and nanotechnology. Today's sessions will continue their scientific journey by focusing on AI-driven biomedical innovations, advanced drug delivery systems, computer vision in healthcare, and translational biomaterials. On behalf of the Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Galgodias University, in collaboration with the University of Cyberjaya, Malaysia, I welcome you all and thank you for your enthusiastic participation. Without further delay, let us begin today's first technical session. It is my great pleasure to introduce our first speaker of the day 3, Dr. Taresh Sarvesh Sharan, Director of AI Research at Origin Medical Research Lab, Bengaluru. Dr. Sharan is an accomplished researcher and AI innovator working at the intersection of artificial intelligence, computer vision, deep learning, and clinical medicine. He leads the development of AI-powered clinical solutions for the fetal medicine, translating advanced machine learning algorithms into real-world healthcare applications. He has earned his PhD from IIT BHU, Varanasi, and has also served as a visiting scientist at Northeastern University. His current research focuses on developing intelligent clinical decision support systems that improve diagnostic accuracy, enhance prenatal healthcare, and expand access to quality fetal medicine. His work exemplifies how cutting-edge AI technologies can be successfully translated into impactful healthcare innovations. Ladies and gentlemen, please join me in extending a warm welcome to Dr. Taresh Sharan. Dr. Sharan, thank you for joining us today. We are delighted
Thank
to
you.
have you with us and we look forward to learning from your expertise. I kindly request you to deliver your expert lecture on your today's topic. The platform is yours.
Uh, Thank you, Dr. Sumedha and Dr. Gaurav Kumar for giving me opportunity and the team to having me for this talk. Let me share the screen first. You can see my screen, I think.
Yes, the screen is visible. The screen is visible. Uh, you can continue. Yes.
So, thanks, Dr. Sumedha, for giving this detailed information about my work. And I'm going to talk more about AI innovation in healthcare as I work in this field. And my session will majorly will be mapped out as like why all of a sudden this change in AI uses in healthcare and all other aspects are increasing. Basic foundational touch on the AI uses and how this is applied at different stuffs, different stages of the healthcare system. Then I'll be talking more about like one case study on how we are using AI in our products in Origin Medical Health uh, to give a quality prenatal care for expecting mothers. And then we will be talking about how this model development goes from initial development to the deployment, and what are the barriers and the challenges that comes in between. What are the regulations and ethics we should follow across across the development platform. And then we will open the stage for the discussion if any questions are there. So, if you think starting off AI is way back 2-3 decades back, but in last 10 or 15 years, the users increased. And around 2015, when the unit and the deep learning kind of models started using getting used in the in the domain, the uses and the product in AI with for the healthcare increased a lot. But if you see in last 2 3 years, this shift from 2015 to 2023 and 23 to 26 is huge.
Sorry,
And
Taresh.
yeah,
Your screen is not visible. Means PPT is not visible. It get blank.
I blanked.
Now it is visible.
If I'm doing slideshow, it's going off.
Now it okay.
You can see
Yes, yes.
the Okay, sorry for that. So yeah, recent 2 3 years, if you think, the uses of AI increased a lot, and that is contributed mainly due to three reasons. We have more data available. The scale of the data increased a lot. The cost of computation decreased a lot, and there is architectural advancement that is beyond uh, the the achievement that is there in last 10 years by introduction of more advanced attention net and transformers and like language models that is there. Why this is important? Because if you see in US itself, around AI in healthcare has a business of around $30 billion, and it's increasing with with a rate of 38%. And in recent years, around 1300 plus uh, uh, softwares and devices is approved by FDA, which is AI enabled. So this gives you a like like very broader vision of how the AI is becoming very important tool and very important role in healthcare improvement across the globe. So going into the foundations of the AI, it's basically like AI is a very huge domain where machine performs some task given with the reasoning that is given for it. And if you see, the machine learning and deep learning are the sub layers, sub domains of it. Uh, Recently, architectures which does the major part in the AI and deep learning evolved so much, and those major parts are CNNs, and then encoder decoder kind of architectures, then transformers and attention, and then foundational models where we have pre-trained weights and pre-trained models, and we just fine tune for our use case that decreases the requirement of edge computing, and so improving the performance. So more like when we say that we are saying that okay, 1300 plus products are developed, which is FDA approved and which are in the clinics. But how these products are distributed across the healthcare system is like, first of all, it is getting used in imaging and diagnostic by for the triaging that if a patient is healthy or some abnormality is there, or to identify the even the tumor, for say in the cases of MRI images or ultrasound, to see if any biometric abnormality is there. So that's the first part where AI is being used. Second part is clinical decision support, where that is extension of imaging and diagnostic, where like okay, we know that particular image is having some abnormality or deviations from a normal path. So based on that, clinicians or experts can take a decision, and based on risk score, and that will help them to better manage the patients like care for it. Then also it is getting used in predictive analytics, all like In imaging and diagnostic, mostly it will be used after a abnormality is occurred, or to identify if the normal and abnormal is there. But along with that, we can use AI to do the predictive analysis. One of the use cases, like for say, we have a like patient history of last 2 to 3 years. We can use that history data to predict like what are the chances of developing abnormality in coming days or weeks or even years. So that is the third part where AI is getting used. Even in language and documentation, AI is getting used. A lot of reporting softwares are there, and like even the note taking for discussion with like within the doctors, while patient management and everything. That at that place also AI is getting used to better summarize and understand like what are the points that should be taken care at the time of patient management. It is also getting used in drug discovery and genomics very vastly. A lot of mathematical models are getting generated and tested. And one thing AI enabled people to better understand, like earlier all the equations for the genomics and the modeling of the any drug delivery is manually fed and ran. Right now, with the introduction of AI, the modeling and the automation of the whole testing and like analysis is automated, and that is increasing the overall speed of research itself. It is also getting used in robotics and surgery to automate it in operations and workflow for, and even in the patient engagement with the monitoring and chat assessment and management also. So when we talk like it's getting used at 8 or 9 places, different places, but where it all started. So AI first reached, as we all know, like first the classification of normal and abnormal, and that's the classic example where, for say, a skin cancer kind of examples were there, where we started to see if a particular image of skin is normal or some abnormality is there, and that's the first place where AI got used in clinics. Then also in the detection right now, even in pathology kind of like task, the AI is getting used, where the more advanced microscopes are equipped with AI softwares, where it takes the images, does the detection if the cells that is seen by the microscope is normal, or what kind of cells that can be seen and all. So that's the second place where it is getting used. And third is segmentation kind of segmentation kind of places where we okay, we know that the abnormality is there in the image, but what is the boundary where all this abnormality lies in the image? For that kind of task, we use segmentation, where each pixel in the image is classified as normal and abnormal, and based on that, we know that okay, this is the boundary where abnormality lies. So these are the first three places where AI got the first reach in overall. Around 3 in 4 of all the FDA authorized AI devices are in radiology and imaging. So you can see that how much extension of AI in radiology and imaging is there right now. But seeing the market, I can also say that in last 2 3 years, uh, the domain is shifting a bit, and not only on the imaging or radiology, even in patient management, or in the reporting side, uh, is also the places where AI is getting used. Okay, so yeah, so here what we can we have a case study, not a case study. It's more kind of like what I work on generally, and I'll give you a brief introduction of why this is very important and how AI can help here to better manage the better manage the uh, whole like whole system, whole health. Uh, uh, s- okay, so think of a case, expecting mother of like second trimester, which is basically like 18 to 22 weeks. Expecting mother is going to a hospital for a regular scan, and right now what happens is a regular scan takes around 40 to 45 minutes to complete. And that's if I give you a range, the the size of the fetus at 18 to 22 weeks or 24 weeks is around I like around 100 centimeters. That's very small. And in that, the sonographer has to scan from head to toe and around 40 biometry 40 anatomies, top to head to toe. That being said, if you think, it is very tedious for the expecting mother to lie down and hold for 45 minutes without moving, so that the sonographer can take all the diagnostic view which is required for proper in proper investigation, if the everything is correct with right with the fetus. On the same time, if you know how sonographers does the scan, is like their one hand will be on the keyboard on the ultrasound, and one on the left hand they will be moving the probe. This gives a lot of strain on the sonographer's shoulders, and a lot of time in US, it's a fact that after 10 years of scanning, generally they retire and they don't do scanning at all due to the ergonomics strain that comes. And in a day, they can only do 5 to 6 scans, because that's the max they can do due to the pain that comes. Along with that, what happens, think of a scenario that a sonographer is doing fourth or fifth scan in a day. After doing 2 3 scans, due to the pain or the fatigue that is coming due to the continuous scanning for 45 minutes for 3 4 patients, what will a lot of time it happens that they miss the diagnostic view for the fetus, and a lot of time that causes false positive or false negative, which may cause in missing of diagnostic values, and that can result into like adverse results in the pregnancy. So that's the scenario behind why we are working on the fetal medicine and prenatal care. So given this scenario, what we do is we are developing a AI end-to-end software, which is a cluster of like 7 8 AI models, along with algorithms and logics, which works in coordination to end to end, to like it captures the images, sees if the diagnostic images are of good quality or not. Once the diagnostic value quality is met, then it sees that all the structures within that diagnostic view is seen or not. If not, flagged out. If it is there, then does the biometry and gives a final output that how much it is within the range or outside the range. Example is for say femur length, for example, or even the humerus. So one of the case I can give you example is like a lot of time what happens is when the fetus is very active, the cord of the fetus wraps around the hand, and that reduces the hand mobility and so the growth. So a lot of time that is the cause why people have like disability in the arms during the birth. So if this software, this whole AI, what will if used properly, it will give you the biometry, yes, but along with that, it can also help you to understand like okay, there is some like abnormality in the biometry, and the cord is wrapping around the femur or the arm of the fetus, and that can affect the overall development of the fetus. And so we should do the like procedure whatever the doctor seems to be adequate for that process, and then they do the process. So the whole aim is to standardize the diagnostic care for mother, so that no false positive or false negative comes in, and uh, basically reducing the like adverse output of the pregnancy, then reducing the fatigue of the sonographer, and reducing the fatigue on the expecting mother. So that's the overall domain on which we work. Okay, so that being said, it's uh, like when we do the whole study, a lot of question comes like like how much we should rely on the AI, and what like a lot of time in the real world, the major challenge comes in like variations are very high, and to cover everything, a lot of things and edge cases we have to take care. But after that also there are chances that something is missed. So we cannot 100% rely on AI, and so one medical person should be there. So keeping that in a mind in our mind, what should we like how this decisions should be taken, and like predictive analytics should be done is like we should use AI for early mornings scores, not for like final decisions, because a lot of time it is correct, but due to any condition it can fail, and that can mm, uh, that can like affect the whole overall outcome. So it is a assistive device. Anything which any software we are developing with the help of AI should be a assistive device, not a diagnostic device, because that will especially in medical domain, because that will affect the whole like uh, to be very frank, it will be like affecting the like life and death situation for a fetus in this case. So it should be always assistive. Then uh, it should be a smart triage, rank incoming cases and work lists, so that the patient which is of most urgent should be seen first, and they like AI can like that's the second application where even the work list or even the patient management can be used with AI, where it can analyze the history, as I was saying earlier, that it can analyze the history and do an predictive analysis that okay, this patient is of high risk, and they should go first for the treatment, or even for the scanning if and based on that we can do the triaging of the patient management also. Okay. Even the resource forecasting, we can for resource forecasting also we can use the AI in a lot of places where we know that, okay, like in a day, 5 patient comes with the scan. So what all is required for it? I- how much of the resources, how much of the healthcare professional, what kind of experts are required. Based on that, uh, we can do the forecasting of the resources itself. Uh, Coming to the recent big change in the AI, which is large language model. Lot of us heard about Claude and ChatGPT and everything, but it is getting used a lot in the healthcare itself also, and like documentation. A lot of people like for say if in the case discussion, generally what happens in hospitals, like 4 5 doctors will sit and they will discuss like how should we process, especially for the critical cases or the complicated cases. They converse that how we should go about this case. Document this AI based this AI based softwares can help to do the documentation and give a proper uh, proper formatted output, like points from each of the doctors or clinicians involved, and then based on that they can take a decision. So it can draft the visit notes. It can summarize the long conversation about a case. It can help to suggest the like codes, write the codes, which codes or even algorithms for AI development, plus also for the documentation completeness checks and all it can be used, and also as a knowledge base. As and when the like complicated cases are coming, it can like we can keep on creating our own knowledge base, and when the new case comes, it can give few it can go through the knowledge base and give you the output of like these are the relevant case which is similar to the current case, and this is the output we did for these cases. This is suggested based on knowledge base, and based on that, it the clinicians can decide like how we can go about the current cases. So uh, the mm, the opportunity that we are looking for AI is like the we have a lot of models that is pre-trained, and we can fine tune where what we do is like for say a lot of people a lot of you would have heard about SAM based models, segment anything model. uh, what people are doing is they are taking these models, uh, getting a small batch of data set of their use case. For say this SAM is not uh, meant to work on the ultrasound images, any kind of ultrasound images. What people are doing that a small data set they are taking from their domain, doing the annotation, maybe 500,000 images. That's very small if you think of healthcare based AI development. 500,000 images, annotate it, fine tune the model, and use it for your use case. It's very fast, low resources required, and the biggest pain of labeling the data is reduced. So uh, that that's the one opportunity we are getting uh, with the availability of this pre-trained models and all. On the other side, we should have we should be cautious. Like these models are can hallucinate because like a lot of time it thinks of a problem statement and generates its own uh, uh, its own thinking, and based on what is available on internet, for example, or even in the database, what are the cases that is available. Based on that, it gives you the output. If that information is not correct, it can hallucinate and divert very badly from what is the intended use. That's why for all the AIs, there should be guardrails and limits in which it should work. Along with that, a lot of these tools are like very narrow and validated not for general reasoners and everything. So the AI is good, but we should have be cautious while using it for the healthcare. It's also reaching the upstream, right from target identification to personalized therapy for each patient we can do with the use of AI in the whole. The biggest problem is in, especially if you ask me, in India, is a lot of published models are there in the academics especially. But if you see, most of them are on the public data set. It gets developed in our academic labs, but it only stays there. Very few of them, two-third of that is even validated on the external real world data set. Half of that, like 50% of the total published literature model are integrated into clinical workflow, and even lesser are getting used in actual settings. So that's the biggest gap that is coming, and we we need to minimize this gap to and transfer this development part from academics to uh, actual setting, actual clinics. There are two kinds of hard problems that is there in AI development. One is on the data quality, bias, and generalization. If you think if we, simple rule on which AI works is garbage in, garbage out. If the data is not adequate for the task, it will not work. No matter how big your data set is, or how much effort we are putting, how good our GPU, nothing will work if the data set is not correct. Second problem is bias. M- like for example, if we are training uh, medical AI on on data set that is only curated in India, and then after approval we are trying to use it in US. The problem is due to ethnicity, due to nationality, everything, like a lot of the variations in the image quality itself is very high. One of the example is, generally it is observation that in India the BMI range goes, for example, in range of 30 to 40 on average. But in the US, it's way higher, and around 60 it is. So this BMI gap can distort the image quality. And then if a AI is developed with a Indian images only, it probability of it failing to US data is very high. So this bias towards a particular type of data can cause a very big problem in the whole AI development. So this bias and equity in the whole data set should be taken care very carefully while developing AI. Also, the distribution shift, even within the same setting or same hospital, or also different sonographers have different. I'm giving the example of sonographer because I'm like it's related to like my work, and ultrasound is my reference. But in general, whoever radiologist or whoever is doing the imaging of the patient, their setting, the machine setting, also differs, like changes the quality of the image. And even if right now, for say, if you take the data from 10 years, from 2015 and to 2025, and train the model, then and deploy it right now, maybe it will work right now, but after few years the machine changed and the quality changed, and then after that again this model may not have that much efficiency on this data. It can work, it cannot work. So this distribution shift, and the term called as data shift, basically, where the all the settings are taken care, but still due to the shift in the data set, like a shift over the period of time, model may work or may not work. So these are the major data set side of challenges that is there. And we, while developing AI for healthcare especially, we should take care to generalize the whole AI for based on these challenges. The other side of the challenge is, okay, we generalize the data set, we train the model which can work on all the geographic l- distribution and ethnicity and everything. But the uh, other side of challenge is when you get the product out, it it should still be explainable why, if it is working, why it is working, if it is not working, why it is not working, right? So that trust and transparency should be there. Also, it should have like automation, like if a person is using it, they should have trust over the machine. Design must be kept like human. Like it should not feel it's very robotic, or so that the sonographers are not willing to use it, and it's very off. Then safety and monitoring, as it in healthcare, it can decide the decisions, so it should be of prime importance that we take care of safety and monitoring, and so the privacy and security. That this is one of the thing we should always keep in mind, privacy and security, because like ideally, no patient data should go outside of a particular health system, even like the hospital premise. It should not go outside due to the different rules that is there. Like de-identification of the patient details, and like consent and everything should be taken care while doing the data set. So these are the things and challenges which is generally there, and we should take care while doing the like AI development for healthcare. So, like challenges are there. So, what should be the evaluation methods? So that's why wherever like we should. One thing is we should always follow the GMLP, that is good machine learning practice. That is for basic ML development, AI ML development part. Along with that, we should always test on the target system. Uh, Always do the calibration based on the machines, or on the requirement, basically, for in clinical setup, generally sensitivity and specificity is more of important, because that relays the true positive and true negative values of the cases. So that should be the measure of how good or bad the the software or the AI is working. Uh, there should be proof that it changes the this like prove it changes decision or outcomes, ideally in the prospective or silent trial. W- when we before we go for even for trial, we should before that we should do a proper study to show that this AI is uh, like on the prospective data, on the real world data with the edge cases, how it is performing, so that there's a trust, and there's people like whoever the users are, they are confident that they can use it. And even after that, if anything comes in, the report should be transparently and adequately documented. So mainly around the globe, three major in context to US, FDA is the one body uh, which which basically uh, approves the medical devices. SAMD we call as software as a medical device, and there are different paths uh, which is used 510(k) or de novo and all. In context to India, basically there are like CDSCO and ABDM, which is Ayushman Bharat Digital Mission, which is in progress. These are in India. The medical devices are not that much very well regulated and still in progress of developing. In the European Union, generally CE is used, and they have their own AI regulation rules which should be followed, and MDRs are very important there. Uh, why India? Why India is very important for healthcare, especially because in if you think India has a lot of variations in the population itself, from north to south, east to west, and along with that, the population itself gives the data set size itself very huge. Recently, ABDM is doing the health ID recording for getting AI ready data, and that is, if you think of like that is going to be like 730 million plus patient data, health IDs. That is very huge, right? And second thing, in why AI is required in context to India is shortage of clinicians that is available at different levels. We generally see in India that metro cities have few centers where most of the people go for uh, uh, good quality of health services. But with the help of AI, this can be decentralized. It can be used at different tier two and other cities also. Uh, if a clinicians, if a expert, or even the midwives, or early career people are there, they can help take a help of AI based uh, softwares to give a higher quality of healthcare to even in the low cost economy systems also. So okay, so these are the AI based challenge developments that is happening. But along with that, a lot of other segments also are there in AI, which on which the developments are going on recently, and those are based on agentic AIs. If you think of for getting getting suggestions and agents which can do interactive analysis based on their skills, based on the their database and the information that they are gathering from the uh, different places. So that's agentic assistants are one place where developments are happening and getting started. Second is multimodal foundation model that is getting developed at different level on the image, on the text, on the different like even on the reports to get better understanding to do the to digitize the health records for the patients and all. So multimodal foundation models are also there. Even the clinical intelligence, where as earlier I was telling that in the scenario where the doctors are doing the case study discussions, this AI can be used to do the documentation and better do a structured outcome of the whole uh, discussion basically. And last one is federated or edge AI. We are developing AI, but uh, running on bedside for a patient, and better on uh, focus, what we called as focus, point of care ultrasound kind of system. If you want to deploy, we need to have these systems very lightweight, and that's where edge computing comes in. So a lot of lightweight model training, lightweight lightweight model training, and like doing the downscaling of the image while maintaining the performance on the same scale. Those kind of developments are also happening. Uh, that's it from my side. We can have a discussion if anyone have any questions. Thank you.
Uh, Hello, is any difficulty, Taresh?
I completed, sir.
Okay, okay.
Uh, we can open it for any questions that is there from the any of the participants or any of you.
Dear participant, if you have any query or any question, you can write in the chat box or you can raise your hand uh, to ask. If you have any query, you can put your concern in the chat box or you can raise your hand. I will unmute you. So there is a question in the chat box.
How
So you can
reliable
take it.
So as I discussed earlier, like AI is reliable, but when it comes to healthcare, we should be cautious, and it depends. If you take care of generalizability of the AI, and if you take care of the proper selection of the target and all other biases and everything, it is well and good to rely on it. But still, you should not take it as a final decision. Still, the clinicians should have a final take on it, and AI should work as assistant. So I think that should answer your question, Pallavi. For Anusha, how AI is useful in research. Uh, Recently, if you think last week only, I got to know I was also exploring, and I got to know that Claude itself developed a tool named Claude Science that is specifically for research part only. So a lot of time. Even in recently, I got into to know that in mathematical modeling for genomics and drug delivery also they are using AI. So I think AI for research in all the places are getting increased and it is best part of AI being used in research is it's increasing the speed of search of the domain, and it's improving improving the quality of the research. Anyone any further questions?
Hello. Hello, Dr. Sharan. Myself,
Hi.
Dr. Lee. So I have one question. Uh,
Yeah.
what this AI and deep learning methods, how they can help in especially diagnosing some important diseases such as the communicable diseases that are present in India. So how can it help there?
Depends. Right now, most as I was talking, most of the uses are in imaging and radiology, and if a disease is based on that, it can help us to identify. But I still feel that it should be assistive, and it should help us to understand uh, like uh, like if it it's very difficult to tell how AI can be very useful in communicable kind of disease, but in general, if for say like tumor kind of disease is there, or any other uh, im- disease is there which is communicable, in those I think AI can help more than like compared to the communicable diseases.
Thank you, Dr. Saran. There is one more question in the chat box. How do regulatory bodies such as CDSCO or the FDA evaluate AI-based medical devices?
Basically, like for any AI-based device to get approved by FDA or any other regulatory bodies, we will have to do a proper study where they will ask for a particular set of questions and set of metrics with the distribution of machine and all the that's why one of the thing we were talking in the slides is generalizability. They want to see how generalizable your AI product is, and based on that, they do the evaluation based on your performance, on the sensitivity in detection or not detection of the abnormalities or the diseases or structures in any aspects in which AI is running. So basically, the study design depends on which kind of AI tool you are developing, and based on that, it is decided.
Thank you, Dr. Sharan.
Thank you.
Thank all the participants for the excellent questions. I thank you, Dr. Sharan, for patiently addressing every query and questions and sharing your valuable experiences in translating AI into real-world healthcare applications. So, as a token of our heartfelt appreciation, we are delighted to present the digital memento and certificate of appreciation to you. I request Dr. Gaurav to do the same, to do the honors.
Am I audible?
Yeah.
Thank you so much, Taresh, for accepting our invitation and delivering such an insightful, informative talk. So, on behalf of Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Golgota University, as well as our collaborator, University of Cyber Jaya, Malaysia, I am honored to offer you a digital memento to you. Kindly accept it.
Thank you, sir. Thank you for giving me opportunity for
Thank you. Thank you so
talk.
much. Thank you.
Thank you, sir. Thank you.
Good morning, Uvanesh.
Good morning, sir.
Hello. Thank you for accepting the invitation.
Thank you. Thanks for inviting, sir.
Ma'am, we can start.
Hello, am I audible?
Yes.
So, it is now my pleasure to introduce our next distinguished speaker, Dr. Uvanesh K, Assistant Professor in the Department of Applied Mechanics at MNNIT, Prayagraj. Dr. Uvanesh completed his bachelor's degree in Biomedical Engineering from Anna University, his master's degree from NIT Rourkela, and obtained his PhD from IIT BHU, Varanasi, where he worked on biosensing devices for studying mammalian cell behavior. His research encompasses drug delivery systems, bioinstrumentation, biosensors, theranostic devices, bioelectronics, AI in biomedical signal processing, medical image analysis, and point-of-care diagnostics. With more than 40 scholarly publications, Dr. Uvanesh has made remarkable contributions to biomedical engineering research, and he currently serves as the program coordinator of the MTech Biomedical Engineering program at MNNIT Prayagraj. We are delighted to welcome you, sir, and thank you for joining us today and accepting the invitation. We kindly request you to deliver your lecture on the drug delivery systems, and the platform is yours now, sir. Thank you.
Thank you. Thanks for the kind introduction. Is the screen visible?
Yes, it is visible. Yes.
Okay, sir. So, good morning, everyone. Basically, in this seminar, I will be more focusing towards intracellular drug delivery system. So, before going into the intracellular drug delivery system, we will have a glimpse about conventional drug delivery system to the advanced intracellular therapeutic. So, drug delivery system has evolved from conventional dosage form to targeted and intracellular system. Many modern therapeutics such as protein delivery, mRNA delivery, siRNA, CRISPR component, or any other biomolecules therapies must enter cell to be efficient. This seminar will explore the principal challenges, technologies that enable intracellular drug delivery, with particular emphasis on photoporation, or we can tell it as we can name it as light mediated poration. So basically, we will see in depth about photoporation. So before discussing into the intracellular drug delivery system, let's see the fundamentals of drug delivery systems. So learning objective. So by the end of this session, the participant will be able to explain the concept and significance of intracellular drug delivery system. They will able to describe the biological barriers involved in the intracellular drug delivery system, differentiate between the conventional carrier and intracellular drug delivery approaches. They can able to explain the principle of transfection and major intracellular delivery technique, including photoporation. So we will get to know about in at the end what I did was like I came up with several papers and we will try to discuss those things. So now we will so the content. So these are the content where briefly we will be talking about conventional drug delivery system to lay the foundation. Then we will move linearly towards the intracellular. So introduction. So drug delivery system is a formulation or a device designed to deliver a therapeutic agent to the desired site at the particular dosage and time, thereby improving treatment efficiency and safety. Conventional drug delivery system primarily target tissue or organ but often fall to deliver large therapeutic molecule across the cell membrane. So modern therapeutics such as protein-based or peptide, mRNA, siRNA, DNA, CRISPR complement enzyme required efficient intracellular delivery to reach their site of action. Therefore, intracellular drug delivery system have emerged to overcome cellular barriers and enable targeted delivery into specific cell and intracellular organelles. Basically, drug delivery has evolved from simply administrating drug to precisely deliver them to the site of action. So, conventional systems are efficient for many small molecular drugs. However, modern therapeutics such as where we talk about protein delivery or something where we need to spear the cell and we are focused to deliver some modified protein or some biological molecule into the cytosol, cell cytosol. So here, when we talk about intracellular drug delivery, what comes into our what stops us in delivering such drug into the cell is that the plasma membrane, which is nothing but it is a selective membrane which allows some molecules to pass into the cell while opposing several molecules to several molecules. For example, if it is small and hydrophilic, the molecules may easily pass. But whereas it is small and hydrophobic, it can't pass. So then comes the polar and non-polar. It may be it may allow the polar and it may not allow the non-polar. These are the some of the characteristics of cellular membrane which opposes the drug molecule or biomolecule to enter the cell cytosol. So introduction. These are the introduction. The cell membrane is a primary biological barrier. So by this we get to know that the cell membrane is a primary biological barrier that limits the intracellular delivery of various types of therapeutic molecule. So what is the difference between drug delivery and intracellular drug delivery? Drug delivery system is a formation of a device that delivers a therapeutic agent to your body while controlling the dose, rate, time, site of drug to improve the efficiency and safety. So whereas intracellular is an advanced drug delivery approach designed to transport therapeutic molecule across the cell membrane and deliver them into a specific intracellular compartment such as cytoplasm, nucleus, mitochondria, or lysosomes, where they exert their therapeutic action. So while conventional drug delivery target tissue or organ, the main difference is that the conventional drug delivery system target tissue or organ, whereas the intracellular drug delivery are specially designed to overcome the plasma membrane, delivery the therapeutic agent again directly into the cell. So the therapeutic agent may be anything. For example, we can deliver mRNA, sRNA, protein, or the edited gene, anything of any size. So objectives of the primary objectives of drug delivery system is to deliver the drug to the desired site of action. It need to provide us with improved drug bioavailability and therapeutic efficiency. Then protect drug from chemical or enzymatic degradation. So reduce systematic toxicity and adverse effect. Provide control or sustain drug release. Improve patient compliance and reduce dose frequency. So what are the objective which isolate the intracellular from the drug delivery system, conventional one? To overcome the cellular, first it is like we are overcoming the cell membrane barrier, delivering the therapeutic agent into the specific target cell. Achieve organelle specific delivery such as nucleus based, nuclear targeted or mitochondria or liposomes. lysosomes as a facilitate efficient intracellular release while preserving drug activity. So endosomal escape. So what is endosomal escape? Many nanoparticles successfully enter the cell through endocytosis, but they become trapped inside the endocytosomes and are later degraded in lysosome. Therefore, the ideal intracellular delivery system should cross the plasma membrane, escape the endosomes, and release the drug into the cytosol. These are the primary objectives of intracellular drug delivery. The primary goal of intracellular is not only to reach the target tissue but also to deliver the successful payload into appropriate intracellular compartment. So now, so already I have discussed. So it is like safety, low cost, and other thing. What are the other ideal characteristics? One such a drug delivery system should have. It should be free, safe, non-toxic, deliver the correct dose accurately, release the drug at a predetermined rate, be biocompatible, biodegradable, easy to administer, improve patient compliance, be stable during dosage, be cost efficient, minimum, it should provide minimum side effect. So these are the various types of drug delivery system. Oncular delivery, nasal delivery, topical delivery, oral delivery, parental, rectal or vaginal. Next, when we talk about injection based on intradermal, intravenous, subcutaneous, intramuscular. These are the various types of drug delivery system, madam. Conventional one. So broadly, what we can do, the conventional drug delivery system such as oral, injectable, pulmonary, and transdermal formulations. So oral drug delivery system where we have a range of formulation, might be like tablets or something. So what we generally do, we encapsulate the formation into an polymeric or for example, gelatin or polymer based matrix. Into that we used to encapsulate it. So upon taking it orally, it goes into our esophagus, then to the stomach, where it as it absorbs the water, it gets swell, and as soon as it gets swell, it gets detached, and the the formulation get released. So inhalation, here it is like we directly take into our nose or mouth. So alveoli epithelia offer get a surface area, especially for lipid soluble drugs. The drug are also excreted by these ways. So it is like directly we used to inhale the formation. So injection-based drug delivery system provide fast systematic effect bypassing the first pass metabolism. Drug can be administered in unconscious or coma state patient. Drug have short half-life, can be infused continuously. So based on the type of drug we are using, it can be like intramuscular way we can inject, or it can be subcutaneous or intradermal. So when we talk about transdermal drug delivery, it is an for homojet is a best example, where it will have different needle or needleless method, where the drug will be encapsulated into a matrix. So when we push it, it get contact with the skin, and there will be like small patches into it which try to pierce our dermal. So based on that, the drug which are attached over there, it get delivered onto our the site of action. It is a non-invasive method or minimal invasive method for delivering medication to skin into the bloodstream. So adhesive patches continuous the drug, containing the drugs are applied onto the skin, and drug crosses the skin surface by diffusion by percutaneous absorption and goes into the systematic circulation. So why now comes like we have seen the conventional where we directly take directly take the medicine. Sir, Gaurav sir.
जी जी बताएं जी
सर 1 मिनट सर. सॉरी.
यस प्लीज. क्या मेरी आवाज नहीं जा रही है युवनेश?
हाँ सर 1 मिनट.
आवाज आ रही है?
1 मिनट सर. 1 मिनट सर प्लीज.
यस यस प्लीज.
कॉल फ्रॉम द होम. அப்பா வர சொல்றியா? இது மட்டும் திரும்ப சொல்லு வண்டிக்கு. Shall I continue?
Yes, yes, yes. Please.
So far the conventional one, it is like it is related to means we used to take the administration the drug the formulated drug, and it will based on it will basically it via the systematic circulation it reaches all the parts of the body. But whereas intercellular drug delivery, it is like more advanced method where we will be targeting the specific site of action. So most modern therapeutic such as DNA delivery, RNA delivery, or protein or peptide acts inside the cell. So the plasma membrane is a primary biological barrier. Conventionally, often fail to deliver macromolecules intracellularly. Therefore, advanced intercellular surgeries are required. So here comes the carrier-based one. Carrier means it is like a cargo. Basically, for example, let's consider a cargo ship or something where we used to transport, for example, now the petrol. So how it is being transported from Saudi, from the west, we used to get the petrol, crude oil from there, and it reaches one of the port. From there to the refineries via cargo ships or cargo containers or trains used to we use. From there it get refined, and from there the petrol reaches us to the local petrol bunk. From there we used to get the petrol. Similarly, here we are using a carrier to uh, to to transport those formulation from one place to another place. The carrier-based drug delivery system is an drug delivery approach in which a therapeutic agent is incorporated into or attached to a carrier material that transport the drug to the desired site of action. The carrier protects the drug from degradation, improves its pharmacokinetics property, enhance the bioavailability, and can enable control or targeted drug release while minimizing the adverse effect. So carrier-based system represent transition from conventional drug delivery to the advanced targeted delivery. By encapsulating or attaching drug molecules to a carrier, we can improve the stability, enhance the enhance the cellular uptake, and increase the therapeutic efficiency. So the objective of carrier-based drug delivery system is to deliver drugs specially to the targeted tissue or organ, improve drug stability against chemical or systematic degradation, enhance bioavailability and therapeutic efficiency, reduce systematic toxicity and adverse effect. Also, it need to provide us with control or sustain drug release, improve patient compliance by reducing dose frequency. So there are like four major subdivision in carrier base, we can say. So first one, it is like liposome based. Liposomes are spherical vascular drug carrier composed of one or more phospholipid bilayer surrounding an aqueous core. Because of these unique structure, they can encapsulate both hydrophilic within the aqueous core and lipophilic drug within the lipid bilayer. It is like the phospholipid bilayer. It is inspired by our plasma membrane. So, liposomes are biocompatible, biodegrade, and capable of reducing drug toxicity while improving therapeutic efficiency through controlled targeted drug delivery. They protect drug from enzymatic degradation, prolong their circulation time, especially when modified with PEG, polyethylene glycol. Liposomes are widely used in delivery of anti-cancer drug, vaccine, antifungal, gene therapy. However, their clinical use can be limited by issues such as high production costs. So, drug leakage and storage instability. These are the main disadvantage with limitation we can tell it for liposomes. So polymeric measles are nano-sized carrier system formed by self-assembled amphiphilic blocks copolymered in an aqueous environment. For example, we use for the synthesis of silicon nanoparticle, we use CTAB. So CTAB, what happened? The soap, it is a surfactant kind of thing. So the silicon molecule go and attach over there. It is like it will have a branched structure on that the silicon nanoparticle will attach. So similarly, like that, upon temperature and pH, the silicon nanoparticle will form based on the arrangements. So, for example, via hexagonal shape we got. So to remove the CTAP, we can use ethanol or something. So ethanol composition. So what happened? The soap solution get out, and we will have a porous structures over there present. So these are the primary use of measles, basically. So they consist of hydrophobic core that solubilized poorly water soluble drug and a hydrophilic outer shell that provides stability and prolonged circulation in the bloodstream. So polymeric measles significantly improves the stability, biosolubility, bioavailability, and targeted delivery of hydrophobic drug while reducing systematic toxicity. Their smaller particle size facilitate penetrated into tumor tissue through enhanced permeability and retention effect, making them particularly useful in cancer therapy, gene delivery, and anti-inflammatory drug. But what limits is that it actually the polymer measles may exhibit limited drug loading capacity, instability upon dilution in the biological fluid. These are the limitations. So, but their smaller size allow efficient penetration into tissue and tumor, make polymer malleable carrier for intracellular delivery. So nano emulsion, basically nano emulsion means here we will be using both the method. Either it will be oil in water or water in oil. So adding and we will mix it up, it becomes a bubble kind of structure when we see those nano emulsion. So nano emulsion or hydrodynamically unstable but kinetically stable dispersion of two immiscible liquids such as oil and water, stabilized by surfactant with a droplet sized physically typically in the range of 20 to 200 nanometer. Their extremely small droplet size provide a large surface area, resulting in enhanced drug solubility and as faster dissolution, provide absorption and increase bioavailability, and it particularly provide poorly water soluble a drug, and nanoemulsion are widely employed in oral or topical, ocular, pulmonary, and parental drug delivery system. They also find extreme application in cosmetic and vaccine formulation. However, the nanoemulsion often required a relatively high concentration of surfactant and may experience physical instability under adverse dose condition. For example, nano emulsion, when we apply it on the skin, sometimes it need to get established based on several temperature also. For example, in India, we experience quite good temperature. So as soon as we apply it, it get dissolved and we can easily apply. Whereas in other countries where the sunlight or the temperature is very low, over there it becomes quite a problem. So although the nanoemulsion primarily enhance drug absorption, they also improve intracellular uptake of lipophilic therapeutic agents. So solid lipid nanoparticles. Solid lipid nanoparticle are colloidal drug delivery system composed of physically compatible lipids that remain solid at both room and body temperature. These drugs are the drugs is dispersed or dissolved within the solid liquid matrix and stabilized by surfactant. So solid lipid nanoparticle combine the advantage of traditional lipid emulsion and polymeric nanoparticle, offering improved drug solubility. So once the improved when the drug get improved, we can achieve controlled drug delivery and sustained release also. So further, it enhances the biocompatibility and reduces the toxicity. Hence, reduces the toxicity. They are particularly stable for delivering lipophilic drug. Lipophilic drug through oral, topical, ocular, pulmonary, or pulmonary system. So SNL also protects sensitive drug from chemical or enzymatic degradation, can be produced on large scale. Nevertheless, challenges such as limited drug loading capacity and drug exposition storage during storage due to lipid crystallization remain areas where still researchers are going on. So SNL combines the advantage of lipid carrier and polymeric nanoparticle. Their excellent biocompatibility and sustained release property make them promising system for intracellular. So these are the summary, we can say, all the four method. While the career based, sorry, hello, நான் திறந்து வச்சிருக்கேன் ஒரு உள்ள ஒன்று வாங்கிட்டு மீட்டிங் வாங்க டிபார்ட்மெண்ட் உள்ள என்கிட்ட ஏன் கேபின்ல So while sorry again. While carrier-based system improves cellular uptake, they must still overcome the plasma membrane and intracellular barrier. To understand how these occurs, we need to first examine the structure of the cell membrane and the biological process involved in cellular uptake. So now, what do you mean by sustained release and control release? So it is a general statement. So sustained release the dosage forms a system that prolong the duration of action by slowing the release of drug, usually at the cost of delaying onset and its pharmaceutical actions. Controlled released are more sophisticated, just simply delaying the release, or designed to deliver the drug at a specific release rate within the predetermined time period. So, biological process involved in drug delivery, targeting, following the administration, the drug frequently passes through the various cell membrane and reaches the target size. So, lower molecular weight drug enter or passes through the cell by simple diffusion. Whereas the macromolecular molecules are unable to enter the cell by simple diffusion process. So, however, the larger power are able to enter cell by active transportation such as endocytosis and exocytosis. Endocytosis, where eat or drink. So inside within and outside out. So both processes require energy. Larger particles are transported across the membrane in bound basis. So endocytosis, the process where a cell absorbs extracellular material by engulfing it with their cellular membrane to form a vessel, which is then pinch off intracellularly. These particles does not pass through the membrane. It is simply engulfed and enclosed. Exocytosis, the reverse process where the material is expelled or secreted from the cell. This is used to rid the waste or secrete the substance produced by the cell. It may be excretion or secretion. So, although the cell possesses natural uptake mechanism such as endocytosis, these processes often result in endosomal trapping and lysosomal degradation of therapeutic molecule. So, to overcome these challenges, various intracellular drug delivery technique have been developed. One such approach is transfection, which enable introduction of nucleic acid and other therapeutic agent into the cell. So, unlike the conventional drug, the nucleic acid cannot rapidly cross the membrane, the plasma membrane, because of their larger size and negative charge. Transfection technique facilitate their entry into the cell, enabling applications such as gene therapeutic, mRNA vaccination, or CRISPR gene editing based. So, transfection is a common method commonly used method for introducing foreign macromolecule such as mRNA, protein, or dye into the cell. And under normal conditions, the cell membrane forms a barrier to prevent this from happening. So these are some example where we have given an example that the cell and it is surrounded by some nucleic acid. So upon transfection, we can able to see the entry of those inhibited nucleic acids. So what is transfection used for? The transfection is a common method to introduce or alter the gene in a recombination cell to study the gene function. The rising popularity of cell and gene therapy is also providing a new use for these technique. Adapting human cell for therapeutic use, these obviously require much more stringent safety standards since altering cell will be reintroduced into the human body. So now comes the various method of transfection. The transfection method can be broadly classified into three approaches: chemical, physical, and biological. Chemical method rely on carrier. Physical method temporarily rely on cell membrane, and biological method use a vital or peptide-based vector. So India here to be precisely discussed here. We can discuss one by one. So first one is a membrane disruption method, where we try to by using inject micro needles or something, where we try to directly inject the vectors, the biological molecule directly into cell cytosol. So which we term it as direct penetration, one among the membrane deposition method. So other one, it is like transit permeable, where using some method like electroporation, optoporation, or sonoporation, we try to rupture the cell for some time. So here the primary focus is to we are trying to deliver some biomolecule to the cell without disturbing or without damaging the cell membrane, so that the same cell can be used further. So what happens when we try to rupture it using physical method? It get ruptured for some nanosecond, and for those within those seconds, we try to deliver whatever target or whatever the formation we wish to deliver. So second one, it is a carrier media. We have detailedly seen those things. So types of transvection, again, it is like biology, chemical, physical, and biological. So comparison, chemical, the efficiency is moderate and toxicity it is low. Further, it is like very low cost and widely used. Viral, it is like efficiency is very high, toxicity leads to moderate level, and it is like huge cost is involved. For example, gene therapy and all. Physical method, the efficiency is very high and toxicity level, it may be from low to moderate. But cost also, it is like moderate, and it is still not in common use. It is like in research and emerging research and development stage. So what makes us to move towards physical? The question. So limitation of chemical method, for example, endosomal trapper, limited transfection ability, carrier associated toxicity. So limitations of viral method, it is like immunogenicity, limited cargo capacity, and safety concerns these are some of the examples. So which lead us to the physical method. Here we does not require any carrier. We does not require anything to carry our pharmaceutical element or molecules, and rapid intracellular access we get, and high cellular viability suitable to difficult to transfect cells or hard to transfer. Basically, here we are going to use the light. For example, if we are using an light, so what happen? We need to have some photosynthesizers, where upon exposing it to light, it absorbs the light, so the energy from the lower band, it will go from the valency band to conduction band. Upon receiving the maximum energy, what happens? So it need to release. So what happened? It try to release those energy in the form of heat, due to which a vapor bubble get generated around those nanoparticle. As soon the nanoparticle which is present nearby the cell vicinity, what happens? The nanobubble, sorry, the vapor bubble gets burst. When it gets burst, what happens? It physically damages the cell for few seconds. As soon as it damages the cell, the molecules which are present in and around around the cell cytosol cell membrane try to enter within those within those microsecond gap. So due to that nanosecond gap, due to that we can using these method, we can selectively deliver those pharmaceutical formulations or bio molecules. So what is photoporation? As I said, photoporation is a laser assist physical transfection technique that uses short laser pulse to create transient pores in the plasma membrane, allowing therapeutic molecule to enter the cytoplasm directly. The optoporation is an emerging intracellular technique that uses laser energy to transiently permealize the plasma membrane. Unlike the chemical membrane, it allows direct cytoplasmic delivery, reduces endoplasmal trapping, and improve intracellular compatibility. So, why poration overcome the plasma membrane barriers, enable carrier-free intracellular delivery, minimize the endosomal entrapment, suitable for hard to transfect cell. So, higher cell viability when optimized. So, the generally what we used to do is that, as I told you, first step is that we used to irradiate a laser and while before irradiating a laser, we need to make sure that we have some particle or some photothermal nanoparticle in around around the cell vicinity. So once we confirm that, when we when we incubated the photothermal nanoparticle, we can irradiate the cell directly. Or else what we can do, we can directly focus a laser light of higher beam, but optimizing it is quite a tough. So second, it is like first step, it is like we can tell it as 1A, 1B. First, before exposing the laser, we need to incubate the photothermal nanoparticle. After incubating the photothermal particle with the cell, what we need to do is we need to irradiate the laser. So when we irradiate the laser, what happens? For example, I synthesize a nanoparticle having 980 nanometer wavelength. So when I exposed that 980 nanometer laser onto that nanoparticle, what happens? For example, after synthesizing, we need to check the absorbance first. So when we check the absorbance, it will provide us what is the nanometer on which the nanoparticle is absorbing the maximum light. So for example, let's consider I have synthesized a nanoparticle having 980 nanometer wavelength absorption. So what happened? After getting confirmation that 980, what I will expose the laser having 980 nanometer. So when we expose the laser having 980 nanometer, the nanoparticle absorbs the laser light. So when it absorbs, the energy transfer happens from a lower energy to higher one, that is a valency band to conduction band. When it reaches the conduction band, the stored energy the absorbed energy get dissipated in and around like a localized heating, which generate vapor nanobubble. The nanobubble expand rapidly. So as it get expand rapidly, when it get burst, what happen? Smaller transit pores forms into the nanobubble due to the mechanical aberration. So, which provide us a path to deliver diffuse the therapeutic molecules into the cytoplasm. So, within few nanoseconds, again the cell membrane reseals it naturally. This is the main advantage of optoporation or photoporation. So unlike electroporation, whereas in electroporation, we will see that unlike electroporation is a method where we will be using maybe high kilo voltage much higher voltage, where the cells which are present nearby the electrode will get poration, which get deliver, which may get perfect deliver, or it may damage the cells. But the cells which are present, basically electroporation, we do it for the floating cells. But here octoporation we can do it for the cells which are dead. And uh, the cells which are present nearby the electrode will get porated, where the delivery will be more or more, while the cells which are present at the center will have, we may have or may not have the delivery during those time of period. So few example is that I have shown here the gold nanoparticle way, where we can able to see the second image. First image, it is a technique where a bubble forms and it ruptures the membrane, which allows the nuclear, the photopath, which allows the biomolecule to enter. And this is then a schematic representative where nanoparticle allow through gold nanoparticle or something, we develop a pores transit upon exposure, the vapor nanobubble get generated. When it get burst, it forms the pores onto the membrane. So similarly, these are the example where the first image, the author used titanium nanoring. Titanium nanoring, it is been formed using lithography method to have the ring shape. After lithography method, the author has using sputtering or thermal, he has evaporated titanium pure metal over there. So he used around 1072 nanometer or something. I don't remember exactly, where he focused laser onto their that rings. So what happened? The rings, on the top of the rings, you can able to see red colors on the first image, where upon exposing it, the heat get generated on those rings, which creates the nanobubble, and the vapor nanobubble. The vapor nanobubble, we in turn create the pores, so that based on the area of exposure, the bubbles get created only where the laser has been exposed, and where we have that titanium nanoring. The other thing is that here the important thing is that we are using nanopulse laser, so it won't directly affect the cells. Only the titanium will facilitate it with the mechanism that it produces the localized heat, due to which the drug get delivered, or the material get delivered. The second image, similarly, the author has created or etched the titanium plate, and yes, the WES came with nano rods, nano halo nano rods, using HF technique, HF and applying the DC voltage of around 60 volt. So selective etching he did, and he tried to pass the similar 1072 nanometer, and he deliver all the cells which are present in and around this area. And similar method, by changing the voltage, the author came up with a flower like structure. We can able to see the the heat generates only at the tip, because as soon as the heat generate, it get accumulated towards the top, and the transportation happen, and the cells which are present at the top, it get the pores get formed, and the molecules which are dispersed in that incubated in the media get delivered into the cytosol. So similarly, on the fourth image, we can able to see here the author used graphene nanoparticle. So graphene nanoparticle, so similar mechanism used. So here on the flexible, to avoid toxicity and other thing, what happened? The author has encapsulated the the graphene nanoparticle into a polymeric matrix, for example, PDMS. So PDMS, and he has created a patch, a micro needle kind of thing. So upon irradiation with a 890 or around 890 or 980, what happened? The patches where and all the microneedle which are present only on the cell creates pores, and it is because of that the inherent property of graphene where it is like thermally conductive in nature. So here all the three are photoelectric. This one is photothermal method. And similarly here the author used nanoparticle of different shape. For example, one has used mushroom shaped one, and other used the microfluidic method. The second one we can able to see the lab on chip 23 one paper, where he or she has used the microfluidic method to fabricate those star-shaped gold nanoparticle. He has used, and using laser, he has successfully created the pores, and he has delivered the biomolecule. Similarly, on the top image, for example, people are now doing these organoid one, where the tumor, we assume that it is like spheroid one. It is like it is it is like particularly it is equivalent to tumor structure or something, where we add cells. We create a droplet of cells, and we reverse this. We create a hanging droplet size. So due to the gravitational effect, what happened? The cell comes at the down, and it creates clumps like structure. So after creating the tumor, the spiral like structure, the author has incubated it with nanoparticle, and upon irradiation, he successfully delivered the variety of biomolecule to the cell cytosol. These are the few examples and latest ones. Similarly, we can use the electrospinning method to have the nanoparticle into it. So you can use it to deliver these things, the different types of. So application, basically high throughput delivery of different payload, including protein, contrast agent, and generic material. High targeted ablation of cell, light targeted ablation of the cell, targeted delivery or ablation of cell in explanted tissue, or even in in vivo. So comparison, electrical, we will see. So electrical, it is like so electroporation, it uses electrical method. Cell viability is moderate, and delivery, it is high when it is at the vicinity of the electrode. Sonoporation, we use ultrasound method, where if we extend it for a longer time, it can rupture the cell entirely. So the viability moderate, and intracellular, it is also moderate, and optimization, it is like very tough here. So magnoporation, here we can use magnetic nanoparticle or mediated one. So magnetic bit. So here the viability is high, but intracellular delivery, it is like moderate. But photoporation, optoporation, we use laser method with the help of nanoparticles. So here the viability is very high, and intracellular efficiency is very high. So before going into other types, let us see the iontophoration, where we will be using positive and negative terminal. We will be using the low voltage method. For example, a patch on the skin, and by applying the voltage, we will try to facilitate those. We will try to stimulate those drug delivery phenomena. So this is the ion operation method. So people came up with inspiring from ion operation and operation. People came up with this structure. Here they used lithographic technique and other selective etching technique. IRE, ion selective reactive ion reactive etching technique to design design a structure like this, which is nothing but a pillar-like structure having a hollow structure in it. It is nothing but silicon. It is fabricated in silicon few silica plates by etching, by lithographic and other technique, etching technique. So when after forming these nano pillar, when we nanofiller, actually the silicon is conductive in nature, so we can use light or electroporation in order to deliver the different sized biomolecules easily. So this was like this is one such method where microneedle or the pillars, we term it as anything. So microneedle or pillar like structure is being designed so that when the cell is incubated, it drift downwards and it adhere to the surface. Once it adhere, it forms something like so it as soon as we apply those light or electroporation, light media optoporation, electroporation on the top of the pillar, the heat gets generated and it creates it ruptures the membrane through which the and though in the specially made way, when the biomolecules are present, it will try to enter into the cytosol. So electroporation, as I said before, we are going to use the higher voltage to rupture the cell membrane. So here we can able to see before applying it is like very intact. When applied, it get ruptured. Here we are going to in the bulk. There are like several methods. One is a bulk electroporation, and other one is like bulk, and another one is poration, and another one is a capillary method where cuvette method where the size get difference. Here bulk, in the bulk we will have suspended cells into it when we apply electricity so of higher voltage. So what happen, the cells which are present nearby get a maximum delivery, whereas on the center it get range, so it is a moderate delivery. So people come up with cuvette based where they try to reduce the gap from 1 mm to much lower, but similar they were like less efficiency. So by inspiring from electroporation of these to reduce those distance, people came up with this technique. We can able to see where the distance it is like 500 nanometer range. So and people try to use the microfluidics method to place those cell selectively or try to optimize it in such a way that at least one electrode will have one cell attached onto it. So the distance get reduced, so the voltage get reduced. So people use 2 to 5 voltage to optimize. Whereas in bulk electroporation, people has used around 50 kilovolt, I think so, 10 to 50 kilovolt. There are several literatures. So as an improvisation to it, he came up with another one where he reduced it to from 500 nanometer to 17 nanometer, and he tried to deliver this the biomolecule. You can able to see the heat generated on the top of these electrode at the tip. So mechanoporation, here we physically try to push our cell into smaller region. So what happened when we try to push to get it stabilized, stabilized or to with the force, what happened? So a few in and around, there were like several transit post creates, and as soon as it come due to the stress, developed localized stress. After when it comes out of that narrow tube, when it has a good stable these things, what happened? The molecules which are present, it try to diffuse into get into the cell cytosol. And here again the optimization is most crucial. So we can, people combine these electroporation, combine these electroporation and mechanoporation for effective for increasing the delivery efficiency. Summary is that to summarize, the conventional drug delivery system efficiency of delivering a drug to tissues efficiency, it delivers drug to tissue but have a limited access to intracellular level. So carrier based system improves the stability but targeting and the cellular uptake. But here also the plasma membrane and endosomal trapping remain major biological barrier. So this gives us a advantage to opt for optoporation or photoporation, where the optoporation or photoporation create transition membrane pores, enabling direct cytoplasmic delivery with higher efficiency and cell viability. So continued advantage in optoporation and nanovara, expanding the potential for intracellular delivery. These are my references which I use. Thank you for your time. And future of drug delivery lies not only in reaching the right tissue, but also in reaching the right cell and the right intracellular target. Hello? Gaurav sir.
Yes, yes, sir. Yes, sir. Participant, if you have any query, you can put in the chat box or you can raise your hand. That's why we can unmute you.
Hello. Thank you, Dr. Uvanesh K, for this wonderful talk. I have one small question for you. Am I audible?
Yes, yes.
So I was thinking like this drug delivery system, how it can be utilized for, let's say, targeting multiple targets for a particular disease system to manage the disease.
Yes, it is quite easy. For example, For example, if you have multiple tumor, for example, one at the right side and one at the left side. So what we can simply do is that we try to have a nanoparticle, maybe encapsulated or something, so which is specific towards that region. So when it goes and reaches over there, we can use our light to get those things delivered. Because when we talk about targeted, we need to make sure that it reaches the right place. So hence I wrote in the last, it is like reaching the right place at the right time and the right way we need to use. I hope I answered you.
Sir, there is another question
Hmm.
in the chat box.
Okay.
Charna Palay, she
So
want to
nanometers.
know about the nano needle.
Sir, actually, nanoneedles are fabricated using lithography technique, basically photolithography or stereolithography technique, and people now use a 3D printers. DLP method. DLP method one, where people use the resin based one to selectively build those nanoneedles. Once you have mask of your interested one, what you can do, you can pour the PDMS with curing, and you can detach those microneedles out. So entire thing, it is like, it is based on how you fabricate those masters. And when we talk about if someone needs those micro needles having the pores, then we need to go for IRE, reactive ion etching technique, and for example, we can go for first we need to coat metal on it, and then metal or something metal basically metal we need to coat. After coating, we need to etch those areas particularly. A costly effort, but it can be successfully done. I hope the nano needle one I answered. So people, for low cost thing, people now uses the resin based 3D methods, where they can achieve up to 20 micro they are climbing, but we can at least we can go up to 30 to 50.
Thank you, sir. Sir, one question from my side. So,
Hmm.
sir, which biomarker are most important for designing the patient-specific drug delivery systems generally?
It is which biomarker? So
Yes,
it's
sir.
up to you, sir. It's up to you. For example, people. For example, if you are targeting the neurological effect, then you need to choose a pharmaceutical ingredient based on those thing. We need to see whether it goes and whether it binds to those site of action or not. That targeting one we need to see, sir. So it is basically up to us. Based on the target, we need to choose.
Okay. Thank you, sir.
Thank you, Dr. Uvanesh, for your insightful responses to the questions and for the enriching session of today's talk. So, may I request now Dr. Gaurav Kumar, Associate Professor and Head, Department of Biotechnology and Bioengineering, Algotes University, and co-convener of the FDP, to present the digital memento virtually to our esteemed speaker.
Thank you, sir. Thanks for your invitation and time. Thank you.
Yes, thank you so much, Uvanesh, for accepting our invitation and delivering such an informative and engaging talk. So on behalf of our department, school and university, kindly accept this digital memento as a token of gratitude and honor from the organizing committee member.
Thank you, sir.
Thank you. So, dear participant, there will be a lunch break, and we will back at 1:30 p.m.
So shall I leave?
Yes, sir. Yes, sir.
I request all the participants to have their lunch and come back at 1:30 to join us for the remaining second half of the today's session. Thank you.